Hi, my name is Ellie Keenan, and I'm entering my research project into the microbiology category. The overarching goal of my project is to gain a better understanding of the mechanisms of the antidepressant fluoxetine through the use of the model organism Caenorhabditis elegans, or C. elegans. Many antidepressants, including fluoxetine, have limitations in their prescription, as we have yet to discover completely how they interact with different genes. Gaining a better understanding of how these drugs interact with certain genotypes can provide insights into whether they will work for individual patients. In my study, I am using the model organism Caenorhabditis elegans, a small nematode worm containing many genes with functional human counterparts. Over 38% of C. elegans protein-coding genes have predicted human orthologs, and 60 to 80% of human genes have an ortholog in C. elegans. For this reason, along with the organism's relatively easy maintenance, C. elegans is used to study a variety of neurological and behavioral functions. My study is centered around a behavioral occurrence in C. elegans called the enhanced slowing response. The enhanced slowing response describes the decreased locomotion of food-deprived worms when they are reintroduced to food. In other words, worms that have been recently deprived of bacteria will slow down when they are brought back to bacteria. The proposed reason for this response is that in food-deprived worms, bacteria triggers the release of serotonin, which then decreases locomotion in the worms. This idea is supported by the fact that in worms lacking key serotonin-related genes, the slowing does not occur. Additionally, the enhanced slowing response is exaggerated by the antidepressant fluoxetine, which is proposed to amplify the serotonin response by blocking its reuptake. My study is designed around the idea that measuring the locomotion of food-deprived worms can provide information about the activity of serotonin and fluoxetine. My research will involve 5 steps, each of which involves the same general method. I am currently in the process of perfecting this trial method before I can run real experimental assays. I've drawn out the method that will be used in my first step, which involves confirming the enhanced slowing response. This method will remain similar throughout all of my steps with minor variations. There are 2 trials, one, this top one, with food-deprived worms, and one, the bottom one, with well-fed worms. Both trials begin with a plate of the worms of the desired strain. In this first step, this is the N2 wild-type strain. First, about 15 worms are transferred using a worm pick into a microwell plate filled with a small amount of M9 buffer. The buffer is pipetted in and out 3 times in order to wash the worms. Then, worms are transferred in a drop of M9 buffer into the next plate. In the food-deprived trial, worms are transferred onto a plate without bacteria. This plate contains a ring of high osmolarity fructose with bromophenyl blue to make it visible, in order to keep the worms from dying on the edge of the plate. The worms are incubated here at room temperature for 30 minutes. Then, worms are again transferred into the M9 buffer and washed before being moved in a drop of buffer to the assay plate. This plate contains a ring of bacteria. 5 minutes after the worms are placed on the plate, their locomotion is measured by counting the number of body bends in a particular worm for 20 seconds. This process is similar for the well-fed worms, here on the bottom. However, these worms are transferred directly from the original plate onto the assay plate to have their locomotion measured. This process will be similar throughout all of my future steps, with the exception of some variations to fit the different objectives. Here I have listed the 5 parts of my experiment. Trial 1 was described on the last slide. Trial 2, which is my second step, will involve confirming that endogenous serotonin mediates the enhanced slowing response by comparing locomotion after food deprivation in wild-type worms and worms lacking the TPH1 gene, which is essential for the production of serotonin. My third step, or Trial 3, will involve confirming that fluoxetine exaggerates the enhanced slowing response by exposing the worms to fluoxetine prior to the trial. I'm most excited for my 4th and 5th steps because they will provide insight into how fluoxetine interacts with certain genes. I'm going to use mutant worms lacking 2 serotonin receptor genes, MOD1 and SER7. I will use the same method as my 3rd step to determine the effect of fluoxetine on the enhanced slowing response of these 2 mutant strains. My hypothesis is that the locomotion of the worms will increase slightly, indicating that the 2 target genes are part of the mechanism of fluoxetine within the worms. Additionally, SER7 has the human ortholog HTR7, which is also a serotonin receptor gene, so the results of this part of my study have direct implications for humans. Hopefully this quick video helped you to better understand the goals and methods of my research project.